So, in this chapter, we're going to cover protists, all things protist. This is our very first lecture on eukaryotes for 1407. What does it mean to be eukaryotic? It has a nucleus, we can't really say it has a nucleus because we're collegiate students. We say it has membrane-bound organelles, okay? So, or its genetic material is maintained or contained within a membrane. So basically that's the fancy way of saying it has a nucleus. The thing about protists is that protists, by and large, are the kingdom of rejects. Everything in the protist kingdom tried to be something and didn't make it, so it was rejected. So when we look at our protists, they fall into three general categories. They are plant-like, which means they tried to be a plant and failed. Fungus-like, tried to be a fungus and failed. Or animal-like, tried to be an animal and failed. So they are the rejects. They have very similar characteristics to animals, plants, or fungi, but they are lacking something or missing something. So as a result, they fall into the protist or kingdom protista. So this is, by and large, my kingdom of rejects. And so instead of classifying them by what they do have, a lot of times you'll see, oh, well, they're lacking a mitochondria, or they're lacking this, or they once in a while, and we will say, like, they have this, or this is a defining characteristic. But if they tried to be a plant and couldn't be, they're probably lacking vascular tissue. And so that would be something that they don't have versus, oh, well, they are multicellular, and they are eukaryotic and photosynthetic, but they don't have vascular tissue. Protist diversity is very similar to bacterial diversity. Bacteria were extremely diverse in the way that they ate, the way they moved, where they could be found, whether they were anaerobic or aerobic. So we see that same type of diversity reflected here. Different types of metabolism, reproductive practices, structure, and habitat. And I basically already said this, but this is the kingdom protista, which is a highly varied eukaryotic kingdom, meaning it's just the catch-all for everything that couldn't fall into a specific category. Or taxon. Let me not say category because we're in biology, we call them taxons. Okay? Protists are not prokaryotic. Number one question a lot of students will miss on this test. What type of cell are protista? And they think because it's pro and pro that they go together. Protists are not prokaryotes, but they're also not fungus. They're also not plants, and they're also not animals. Okay? So they are just rejects. When we talk about fungus, they have characteristics of fungi, but protists are motile. And with the T, does anybody know what motile with the T means? It's different than mobile with the B. Motile with a T tells you that it has something to do with an aqueous environment. It might be water or something fluid, liquid-like. So it's swimming. Fungi aren't motile. So it has characteristics of fungus, but they're not fungi because they can move. They're undifferentiated, and they have no embryo protection. Well, plants are differentiated, which means that they have specific specializations, and we put a seed coat on our embryo, so we have seeds. So it acts like a plant, yet it's missing some characteristics that plants normally have. When we're talking about the animal-like protists, they're undifferentiated, and again, have you guys heard this term undifferentiated, or do I need to explain it? Differentiated means that you have in your body, your cells are differentiated. Your skin cells act as skin cells. Your liver cells act as liver cells. When you are undifferentiated, everything acts the same. Does that make sense? So whenever I initially teach this, well, of course, I'm teaching it to people who are of the same age, but I would say, how many of you are going to graduate in 2021? And then you would raise your hand because, okay, yes, I'm going to graduate in 2021. Now, how many of you in 5 years are still going to be the seniors of 2021. You're not. You will always be the class of 2021 because you all started from the same. But hopefully in 5 years, some of you are doing different things. Some of you may be teachers, some of you may be in medical school, some of you may be janitors, some of you may be whatever. But you've differentiated. You've become specialized. You started from the same beginning, if we're talking about graduating, and then you've gone off and done your own thing. You've differentiated. When cells are undifferentiated, there's no distinguishing of duties, or there's no delegation of duties. They all do the same thing. In animals, we clearly know that animals are differentiated. You and I are a perfect example of that. But these protists have no complex development. We have complex development. No collagen. You and I have tons of collagen, and as do most animals. So that's unlike animals. So again, it's telling you what they don't have as a form of classification. So slime mold is an example of protists, algae, green algae and brown algae. Ciliates are a type of protist. So these are just examples out of your textbook of protists. Now, you don't need to know this. There are a ton of rejects, and you can see that clearly. We are going to look at the large groupings, okay, and we are going to talk about the defining characteristics, give a couple of examples of each, and then move on. So you're going to be responsible for what's on the slides. This is one of those things where I told you memorization is going to be key. So you come up with each of the groups and the characteristics that go with it, and if there's any accompanying examples, and you memorize that, okay? So we'll get halfway through today. I think we're going to get through heteroconts, and then we'll do Cercozoa, amoebozoa, and opisthocons, and Archaeoplastida next week. Okay, so we'll get this far today. Still talking about how protists are diverse. As far as metabolism, it says that they're aerobic. What does that mean? They have to have access to oxygen, but they could be heterotrophic or autotrophic. You know the difference or the distinguishing characteristics there. Their reproduction methods could be sexual or asexual. It just depends on what type of protist it is. We will not study protist life cycles, but there are, like we'll look at plasmodium and you can tell the difference. You won't need to, but they're there. As far as structure, here's one of the things that keeps it away from being an animal or a plant. Some protists are unicellular. What does that mean? They have one cell. Animals are all multicellular. Plants are all multicellular. So when you show up as a unicellular wannabe plant, you're immediately kicked out because all plants are multicellular. Now then it says, or they grow in colonies. And we've discussed the term colonies when we reference bacteria. Do you remember what I said about colonies? Anything about colonies? Right. So they grow, it's a bunch of the same organism, a bunch of the same cell that all grow together. And they're just all hanging out. It's just like one large hangout. But what we began to see in certain organisms, not necessarily all protists, but when they grew in a colony, some of them said, hey, I'll take care of protection, and this group right here will take care of eating. Now, when you take care of protection or you take care of eating, that's differentiation. So not necessarily are we going to see that here, but unicellular organisms tend to grow in colonies. Okay, and that's for protection purposes and access to resources. But look here, little differentiation. So, I mentioned that, but then again, it's written there. Characteristics that we may see in protists, a contractile vacuole. So, we've talked about vacuoles before. Plant cells have large central vacuoles. Some animal cells have vacuoles, but usually vacuoles are used to store water and nutrients. But a contractile vacuole is one that when it fills up, it will squeeze itself. It contracts. And it's so that it can get rid of its contents. The contents or the contraction of that vacuole have two purposes. First, to maintain homeostasis. Because if that organism, because most protists are aquatic protists, live in aqueous environments, marine, freshwater environments, if they take in too much water, they'll swell up and burst. So the contractile vacuole will fill, and then they can squeeze it and get rid of that water or whatever fluid is that they have. The second purpose of a contractile vacuole may be to aid in movement. So whenever they squirt its contents out, it kind of propels them a little bit through their environment. So it could aid in movement as well. So a contractile vacuole helps to maintain homeostasis so it doesn't take on too much water, and then as a result, it burst, or to help with movement. Protists have food vacuoles. So maybe not exactly a mouth, but a vacuole that takes in food and helps to break it down. A pellicle, which is just a supporting layer under the plasma membrane. So this is more inside of the cell. Pseudopods or pseudopodia, these are false feet used for movement, and we kind of talked about them briefly in 1406. We'll talk about them again when we get to amoeba next week. Flagella, most people are familiar with flagella. This is for long distance movement, and cilia are for localized movement. So flagella are usually for long distance movement, and cilia are for localized movement. Here's an example of a ciliate. We'll talk about these groupings. But this is considered a ciliate because it has a bunch of cilia surrounding it. Characteristics of ciliates, how far do you think they move? I'm a ciliate. You've actually studied this one, but still, just in general, this falls in the grouping of ciliates. They're classified as ciliates because they have all these tiny cilia. What does that mean as far as their movement? They stay pretty local. Okay, we have a contractile vacuole here, and it says that this is emptied, which means it has just released all those fluids so that in order to maintain homeostasis. You're going to use cilia to move in aquatic environments, okay, to kind of swim around. But nonetheless, you can tell that this is semi-organized. So you've got membrane-bound structures, so it's clearly a eukaryotic cell. Here is this organism that you studied in 1406 and now again in 1407. This is the paramecium. A paramecium is a ciliate, so an organism that is covered with cilia on the surface. It kind of labels macro versus micronucleus. These are characteristics you'll see coming up as well. The macronucleus really just directs all the metabolic activity, and the micronucleus holds the genetic material. Okay, so macro kind of conducts the metabolic activity for that organism. The micronucleus houses the genetic material. There is a term, the trichocyst. The trichocyst is, I think they're pretty cool. They're little stingers. They reference them as being kind of like harpoons. We see these similar structures in more complex organisms, but it's a defensive mechanism. So if it feels threatened, it can eject these trichocysts. But you know what they kind of look like? This is probably maybe you guys have no idea what I'm talking about. But you know those cork guns that, like, old school? Like, you pulled it and it had the cork, and when you shot it, it shot the cork off, but the cork had a string attached to it, and then you could, like, pull it back. See, maybe I'm way older than you guys. That's what it's like. So, like, if it feels threatened or it needs it for a defensive mechanism, it will shoot out that little trichocyst, that harpoon. It may or may not have a toxin in it, but it'll kind of poke at or stab, but it's also unicellular. But nonetheless, it pokes at or stabs something, and then it kind of winds it back in so that it can use it again. Okay, so it has a string attached to it. They're pretty cool. But that's also coming from a nerd, that they're pretty cool. Protist habitats. Again, we've already talked about the fact that their habitats tend to be pretty diverse, but Generally speaking, you're going to find them in aquatic environments. You're not really going to find protists out on dry, arid areas, in dry, arid areas. Phyto, what does phyto tell you? Plant. So phytoplankton is a photosynthetic protist. Okay, and most of us are familiar with the fact that if you say plankton, that we're talking about that it's found in water. Not like SpongeBob, but like plankton, although he is a phytoplankton. Not SpongeBob, but plankton. Yeah, okay. Just wanted to make sure I kept that clear. What does zoo mean, or zoo? Animal. So this is an animal-like plankton. So we know that it is not photosynthetic because it's not phyto, so it's animal-like, so it's heterotrophic, probably feeding on those phytoplankton. Detrital or detritus feeders. These are detrivores. What does that mean that they eat? Dead or decaying matter, yes. And then parasites are those that require a host of some sort in order to maintain their metabolic activities. All right, here are the groups that were on that one slide, and this is just the intro to it. So, like I mentioned, I would go with each of the groups and then their characteristics. So I would start maybe, I don't know if you're making a chart or if you're making flashcards, whatever it is that you're doing, but your first grouping is excavates. And excavates, their main defining characteristic is that they lack mitochondria, so that would be one of the very first things. When we cover that here in just a few slides, I'll give you a few more details and give you examples, okay? And then we'll go on to the next one. The Discae cristata, meaning the cristae are what we find inside of the mitochondria. They're the little folds inside of the mitochondria. When we have these I'm not sure why I can't say it, but anyways, when we have these organisms, they have what we call a kinetoplast, and that's just kind of like a really long mitochondria with a small structure on the end, and I'll show you what that looks like. The alveolates, they basically have a bunch of cytoplasmic structures, but they use flagella or cilia to move. This is a kind of course, a diverse group, as they all are. Heteroconts, we start brown algae next week, but heteroconts are the largest. This includes brown algae growing up to being like 200 meters in size, but requires aquatic environment. So as soon as you take it out of water, it's just this big blob because it has no type of reinforcement. The cercozoa include our amoebas. Okay, the amoebozoa include amoebas and some slime mold, but they're going to be differentiated by their movement types, and these have filamentous pseudopods, and this has cytoplasmic streaming, but we will get there. The Archaeplastida, this includes red and green algae because heteroconts are brown algae, so you start to learn or memorize these. These are believed to be the ancestors to plants, that the Archaeplastida led to the development of plants over time. And then we have the opisthecons. The opisthecons are believed to be the ancestor to animals. So in the protist category of rejects, we have animal-like protists, plant-like protists, and fungal-like protists. Of those groups of protists, there's a group of plant-like protists that are believed to be the ancestors of plants, the actual eukaryotic plant. And the opisthecons are believed to be the ancestor of animals. Okay? When it says here, in several protist groups, plastids, first of all, do you remember what a plastid is? Yes, just a little bit of genetic material that kind of got picked up. Okay, it's not part of the main genetic material, it's just a little bit that kind of got picked up. Plastids evolved from endosymbionts. What are endosymbionts? What is symbiont? That was a relationship. An endosymbiont means that there's a relationship between two organisms in which one actually inhabits another one. So it lives inside of the other one. So, of course, if it's living inside, it could leave part of its genetic material result in a plastid. All right, first group, excavates. And as I mentioned a moment ago, their main characteristic is that they lack mitochondria. They lack mitochondria. They are flagellated and they are single-celled. They're unicellular. Generally speaking, this group is adapted to parasitism and can live in areas of low oxygen. Can you tell me how the fact that they lack mitochondria might kind of work well with low oxygenated environments? Mitochondria require oxygen in order to make ATP. So if I don't have mitochondria, I don't need high levels of oxygen. But if I'm a parasite, where am I getting my energy from to begin with? My host. So you kind of see how that all fits together. Okay? Diplo, what does Diplo tell you? 2. So Diplomodadida, they have 2 nuclei, and they have flagella, which we already know. This includes Giardia. Usually when we have 2 nuclei, that's a macro and a micro. The macro is taking care of metabolic functions. The micro is my genetic material. Okay, so just so you see that. Giardia is, whenever they say don't drink the water, this is usually the protist that's associated with that diarrhea that comes along with it. Okay, so another thing to point out is we don't really use, when we don't have to worry about antiviral when we're dealing with protists. If you have an infection caused by a protist, what can you do to treat that protist? You can use some type of antibiotic. Why? Because it's alive. You can kill it. So having a protal infection, not that it's exciting, but if it's not too far developed, you a lot of times you can get rid of it. The parabasala. This is talking about the basal layer. So you see basal there. It has an undulating membrane. And I'll show you a diagram of it so you can kind of see. But it's just kind of, it doesn't have specifically a flagella. It just kind of like moves through water. It's or not necessarily water, but an aqueous solution. It's pretty cool looking, but bad deal. This is what causes trick or trichomonas vaginalis, which can inhabit itself in a male or a female. Kind of look at that. So here's Giardia. Giardia, and it's showing you here, even this example is provided from fecal contamination. So where did it come from? Poop. All right, and I told you that usually causes a bunch of diarrhea when they say don't drink the water. That's an issue. That protist infects you. So if you know that that's you, like a lot of people who travel out of the United States and drink contaminated water, they come back and their stomach hurts, they go to the doctor, and sometimes they've picked up something by drinking that water. So that it's pretty much a rule when you visit a foreign country that you drink water from water bottles, yes. Just to be on the safe side. And people are like, oh, you're fancy or you're a snob. No, I just don't want to poop myself to death. Okay, that's all it is. So trichomonas vaginalis, these are, this is the protist that causes like UTIs and like vaginal infections in men, or not vaginal infections in men, vaginal infections in women, but penile infections in men. Usually accompanied with it burns when I pee, discomfort in the lower back as it continues to spread. So a lot of people just think they're tired. Women think that they're about to start their period, but in actuality they do have an infection. In both instances, you can take an oral medication and have that one cleared up. So I've already said this before, but anytime you notice something different about yourself, you should go to the doctor, because there's a good chance, even if you're worried about an STD, that it might be something you can get rid of. Okay, hopefully just pray it's not viral. Discicristates, or cristates. This is talking about, again, I mentioned, the organization of the mitochondria. Inside of the mitochondria, when we drew it in 1406, I don't know if you remember, but we drew the mitochondria, and then we did like this. These are cristae. Okay, so what we see here is it has a different organization inside of that mitochondria. All right, so it mentions here they have a disc-shaped mitochondrial cristae, and they are also motile by means of a flagella. So moving in aquatic environments using a flagella. Euglena, you guys have already been exposed to this. It's in the lab that you have now. We used it in 1406. But this disci cristae is free-living and photosynthetic, yet heterotrophic. Photosynthetic, yet heterotrophic. So unique characteristics. And then we have the Neato Plastics. Neato Plastics are the ones that have the really long mitochondria with that small modification on the end, and I'll show you what I'm talking about. So what a Neato Plast is, I mentioned, first of all, it's within that mitochondria, it has this it's really kind of elongated, and it has this contraption at the end. This extension or modification is just this large genetic protein, so DNA protein. And again, I'll show you a diagram. They use an undulating membrane to move. Heterotrophic cells, most of these are what we reference as animal parasites. Trympanosoa is it is an example of a neatoplast, but here it is in different parts of the world. So this is the same sickness, just in different parts of the world. African sleeping sickness is going to attack, and most of the time this kills. It's transmitted by the tsetse fly. This is very prevalent in Africa, of course, which is why it's called African sleeping sickness. You get bit by this tsetse fly, and you may be asymptomatic for a while, meaning you don't show symptoms, but then eventually it starts to deteriorate your nervous system. And then these individuals expire if they don't have access to medical care. Chagas disease is similar in its pathophysiology and the way it infects. But the thing is that in America, when we start feeling bad, and we have access to medical care, and what can we do? Treat it and get rid of it, right? So again, it may be asymptomatic for a while. We see in a lot of kids that their eyes swell up and get really big. And because the symptoms are similar to other sicknesses, not anybody normally thinks, oh, this could be super bad. But that's why you go to the doctor, and you're like, I'm not really sure why their eye is doing this. But it can be treated. If it's not treated in all cases, this organism can overcome you and cause organ failure, so it will kill you. See the exact same thing in leishmaniasis. So this is just found in a different part of the world. Here is the euglena, which you've looked at, and there's a model in our lab that we're using for lab. So you can see the complexity. Notice that it's a eukaryotic organism. See its chloroplast, so you know that it has photosynthetic capabilities, and so on. All of these, I'm sure you know, but you have access to these notes, and there's videos in here that give you like diagrams and simulations for some different experiments, so you can always click on those. I just don't do it while we are in lecture. Here is the neatoplast. Look at this mitochondria, first of all. This really elongated mitochondria. Very unique. And then you have the neatoplast, which is that DNA protein, that mass of a DNA protein. Unique characteristic. Again, you can see, I love this picture. Not the organism, but just the picture. How close it is. The size of a red blood cell, you and I can't see these with the naked eye. We have some in focus over in the lab next door whenever you're looking at the plasmodium. It's a red blood cell slide. But look at how tiny this organism is, yet how complex in its structure. So pretty cool, and this is how it moves. So it's an undulating membrane. The alveolates. The alveolates can be broken up into a couple groups, and we're going to talk about each group. We have ciliates, and you and I have already, when we talked about the paramecium, I said this is a ciliate because it has cilia. So that's that characteristic there. Dinoflagellates, and then apicomplexans, or apicomplexa. We're going to talk about each, so this is just the intro slide to the alveolates. Beginning with ciliates, also called cilophora. They use cilia to swim or to move, so it's really localized movement. Again, they have two types of nuclei, the micro and the macronucleus. Once again, the macronucleus, usually dictates the metabolic activities. The micronucleus is used for genetic material, is their actual house genetic material. Okay? And this is the exact same organism that I showed you on a previous slide, the paramecium. Okay? Just so you know that. These do have the ability to conjugate. Conjugation is a form of sexual reproduction. So we can have genetic variation here on these organisms. And they can make trichocysts. Trichocysts were that what I pointed out to you. They kind of functioned like that pop gun or like a harpoon that can work as a defensive mechanism. Okay, again, there is an animation here that kind of goes through and helps you understand the structure a little bit more, but macro versus micronucleus, you can see that. Dinoflagellates. Dinoflagellates, and there's one other group that we'll look at next week. These are really pretty, but they can be very toxic, and so you just kind of have to stay away from them. Whenever, I don't know how many of you have ever been on the ocean or been in a glass bottom boat. A glass bottom boat is like a tour type of thing that they can take tourists on, and you pay $20 and go. And you're on a boat that has a glass bottom so you can see the bottom of the ocean. And it's pretty cool depending on the time of day. But there's always a lot of sparkly things that look like crystals and jewels, and those are these dinoflagellates. They look really, really pretty. But depending on what species they are and where they're located, they're extremely toxic. So you stay away from them. So they look pretty. We have some, of course, next door that you're looking at in your lab. But the dinoflagellates, they have a shell, and the shell is something that they secrete metabolically. Like it's a shell, and they also call them a test. We'll call them test in other organisms. It's just a hard external casing. So, but they're super pretty. A shell of cellulose plates. Do you recall what cellulose is? It is a carbohydrate found in plants and their cell wall that you and I cannot digest. Okay, and I talked about that in previous lectures. I don't think this semester. But the fact that it can't be digested is why you cannot digest plants. They swim using flagella, primarily marine, and marine tells you specifically I find them in salt water. Some are photosynthetic, and so they're photosynthetic and they're marine. So they're oceanic primary producers. Okay, releasing oxygen. And notice it just says some, it doesn't say all. Some are bioluminescent, which makes them look really, really pretty. And everybody wants to touch them. And some cause red tides. And red tides is on this next slide. I'll show you what it says. Yes. Red tides are when the dinoflagellate blooms, it releases a neurotoxin into the water. And it kind of dyes the water red, but it also kills a lot of the organisms in the water in that area because it is now been infected with a neurotoxin. It doesn't kill shellfish, though. So any shellfish living in that area might pick up this toxin. However, any organism that consumes the shellfish will then be impacted by that neurotoxin. So there are times when you don't eat shellfish. Is that a question, Rachel? This one doesn't necessarily help another organism thrive. This is just an example of a dinoflagellate. So dinoflagellates are very diverse. Some of them are photosynthetic and are a good part of the ecosystem, and some of them release toxins. So whenever you go through and study them, because Protists is a kingdom that we have to study in 1407, you talk about just the things that I know are going to stick out in your head about these different groups of Protists. Does that make sense? So overall, like this one specifically, it looks pretty, and when it's not in season or blooming, it's just a part of the ecosystem. However, when it does bloom, it will release that toxin in this area. Look, here's kind of what that looks like as that water is infected with that toxin. And it's called the red tide because it looks red. But does that kind of answer your question? Like, why are they important? Like, impact the ecosystem? That's a great question. This species specifically, I can't answer, confirm or deny that. Dinoflagellates in general, the photosynthetic ones, because they do function as a producer for the ocean, and the ocean is where we get the majority of oxygen from, that would be an issue. But specifically, the ones that cause the red tide, maybe not. I don't want to say, oh, for sure it's not a big deal, because it may be. And I just don't understand their importance to the extent that I need to. But I do understand overall, like, this specific one. Maybe not. So again, what I had already mentioned is the fact that they do release that neurotoxin. And for shellfish, it doesn't necessarily kill them, but because they're filter feeders, they fill up in it, and it gets in their tissue, so that when an organism consumes that shellfish, that becomes an issue. Okay, and it's poisonous to the organism who consumes it. Apicomplexa or apicomplexans are non-motile, meaning what? Non-motile. They don't move in aquatic environments. Motile means that they move in aquatic environments. Non-motile means they don't. Animal parasites. The example of this is malaria or plasmodium, which causes malaria, and the disease which you and I call malaria. Okay, so they themselves do not move. They'll move within fluids, meaning if they're there and the fluid is flowing, they flow with the fluid. But they don't swim or have an extension that helps them to do so. Okay? As far as their reproductive cycle, they are both asexual and sexual. This is also explained in a great detail in your lab. You do need to be familiar with plasmodium and the life cycle for malaria. Here is very similar to what is in your lab book. So malaria starts off in a female mosquito. And the female begins to make sporocytes. So sporocytes are these haploid gametes that eventually will make their way to the mosquito's salivary glands. Okay? They don't necessarily have a reproductive process just like ours. But if they're in the salivary glands, when that mosquito bites a regular, uninfected organism, it will secrete whatever is in its salivary glands into that organism through its needle, or through that needle that it pinches or pierces with. Well, so then it puts these sporocytes inside of this unexpecting human. They go directly to the human liver, where they begin to replicate and now go from just being female gametes, some of them differentiate into male gametes, and they have the ability to take over the red blood cells and then sickle those cells. When so now we have male and female gametes. They're released into the bloodstream. They begin to host on the red blood cells of that organism. Initially, symptoms of malaria are like, I'm running a fever, I'm cold. So do they think they have malaria? No, what do they usually think they have? The flu, something like that. And so they wait it out for a little bit, and because it's a living organism, and our body will, of course, try to fight it, but it's attacking our red blood cells, it's far more aggressive than the flu. And then, of course, it mentions that in the event that another female comes and bites that infected human, whenever it sucks up the blood, it's now carrying both male and female gametes, so it's more aggressive to the next person that mosquito bites. Okay? And it mentions this in this cycle, and it talks about it in your lab. And we have a poster in the lab. We also have it in your text and in your lab book. So plasmodium. Here's a video that also discusses it in your notes. All right, heteroconts is where you and I will pick up on Monday. Any questions here?